So this is a recording of the review of study guide for exam 2 covering chapters 3 and 5 of our course material. And on your blackboard you would find a blank version of this study guide, and during this session we will cover any gaps that I have left there. And typically if there are gaps, those are important concepts or terms that I really want you to be familiar with. So they're always worth of paying attention. And you'll also notice that I have provided answers for some of the gaps already ahead of the time. Those are written in black. Usually if you have to fill in something, that would be highlighted on this version on a blue color. So let's make a start and have a look of the chapter 3 where we discussed about biological macromolecules. So on this chapter we really focused on 4 types of molecules that you typically see in nature. But before we dived into those, we started with some general concepts. And one of the general concepts you are already familiar from the past, that was the definition for the molecule. And we saw that a molecule, when we had 2 or more atoms, and these atoms were bound together, and these bindings between the atoms were by chemical bonds, and there were various different kinds of chemical bonds that we could see. Another thing that we saw, that in the molecules that we had a look of initially, the molecules were typically electrically neutral. And as the introductory part, or kind of setting the scene part, we also had a look of 2 ways of building and breaking down molecules. Before we did that, we had to highlight an important concept, that there are different number of units that we can have for a molecule. If we only had a single unit, so that's shown in this model that I've included here as this sugar molecule that I've added here. This would be a single sugar molecule. If that's the case, we talk about a monomer. So mono means 1. So 1 unit only that we're seeing here. If we have multiple units, as you can see here, and this number of units could be anything really from 2, 3, 4, all the way to however large, connected to one after another. This would be in this case known as polymer. So poly referring to the term many. So here we have a polymer with multiple units together. And the 2 processes where we could form or break down different kinds of combinations of polymers or monomers were dehydration, synthesis, and hydrolysis. So let's have a look of that. And we're going to start with the dehydration synthesis. So what happens in dehydration synthesis? It's always a good idea to have a look of the term that we're really using. That usually gives us a clue. And let's pay attention first to the term synthesis. When we're synthesizing something, we're building something, we're making something. So in dehydration synthesis, what we are trying to do, we're trying to build a polymer. And as you see here, this polymer is made of 3 units that have all joined together, but the number of units could be really anything at all. And you'll see that initially these were simple monomers, which were then combined together to form a polymer. So that's the synthesis part of this term for this process. What else can we learn about the name of this process? Let's pay attention to the term hydration. Hydration refers to always to water, and if it's a dehydration, that gives us a clue that now water is being lost. And we actually see it here. This is the water molecule that is lost as a part of the process of forming a polymer. So we take individual monomers here and we join them together to form a polymer here. And as a byproduct, as we can see here, we're forming water. And the number of water molecules that we might be left with, well, that would always vary depending on the size of the polymer that we're forming. How many monomers are we joining together? So the term dehydration synthesis really tells us what's happening in this process. So I strongly encourage you to always take a little bit of time and trying to figure out that what are we really looking when we're talking about different kinds of processes, what we're seeing. And we do that by paying attention to the terms that we're talking about. Well, next I want to talk with you a little bit about the other kind of an option of processes that we keep running. And on this option, we are actually taking a polymer, as you can see here, polymer made of 3 subunits that are all connected to each other. So we take this polymer and we break it down into individual monomers. So in this process, we're actually taking something that's a larger concept and breaking it into smaller subunits. And to achieve this, we also need to do something extra. So the key here is to remember that when we're talking about lysis, lysis is usually referring to some sort of a breaking apart, something is no longer together. So in this case, we're breaking apart the polymer into monomers. And let's look at the other part of the name of this process. There we see again this term hydro. Well, the term hydro in this context tells us that to break this polymer into monomers, we actually need water. That's essential for that to happen. So we're using water to break apart these bonds between the polymers that we see here, and they break up into monomers. So on this one, we were talking about hydrolysis. We're breaking something apart with the help of water. So those were the 2 basic processes that we needed to know in this chapter. Let's see what else we might want to know. What other concepts might become important for us as part of this chapter? Well, this next one that I really do want to pay a little bit of time when we look at it is going to be the concept of enzymes. So we noticed that your textbook gave a little bit of a different definition, and I think that this is sometimes a little problematic because we want to be careful that we use correct definitions. But enzymes helped with chemical reactions. And remember that there can be many different kinds of ways how this help or assisting happens. But the key thing here is that this reaction would happen regardless. No matter what we do, this reaction would regardless take place. So let's take an example. And in this example, we're looking at this block here. This block in this chemical reaction would normally get to be broken into two parts. This would happen regardless whether we have an enzyme or not. It would be broken in two parts. But now that we recruit an enzyme to assist us in this reaction, what we notice is that this enzyme helps or assists this reaction to happen. So this enzyme makes this reaction to happen. It could be faster, it could be more efficiently, it could be with lesser amount of energy needed. There could be many different ways how it assists. But the key message here is that this reaction would happen anyways. An enzyme plays a role of an assistant in making this reaction happen. Another important point to notice on this diagram that I've kind of provided here to explain what an enzyme does is that our particle, in this case that would normally be broken into two parts, binds into this enzyme to its active site. So we see that it has a site here where this component was able to bind. And as a result, the shape of the enzyme actually changed. If you pay attention to this bottom part of the enzyme. And once the enzyme has assisted on making this reaction happen, the enzyme returns to its exact original shape. So enzyme is not spent as part of this process. Enzyme can be reused time and time and time again. So enzyme really does not get used at all as part of these reactions. It can be reused. So that was our definition of enzyme. Enzymes assisted in chemical reactions. And like I said, the assistance could be many different ways. And in this example, we see how the enzyme can bring down the activation energy needed for something to happen. So on the x-axis, we would see basically the reaction, the progress of the reaction. You can consider it also as if it would show us the time, how things progress over time. And on the y-axis, we see the energy needed. So in a normal situation, we would have this reaction requiring a certain amount of energy. And in this case, it would be all the energy reaching this line here. And once the energy of the reaction reaches this line, the reaction proceeds. So that's what would happen normally. Now, when we add enzyme, we're actually able to bring down the amount of energy needed. So the enzyme helps us to bring down the activation energy needed. And now that it's lower activation energy than what was needed initially, this reaction will take place with less energy required for it to start. So you'll notice in this chemical reaction, the role of an enzyme was simply to make this reaction happen with lesser energy having been needed. There's a bunch of examples that I've listed there: lactase, trypsin, maltase, sucrase. And one type of an enzyme that we often see, and I think that your textbook really used to highlight, would be a catalyst. Catalysts are enzymes that speed up reactions. So not all enzymes speed up reactions. It's especially catalysts that serve this purpose. Catalysts speed up chemical reactions. So that's just one example of many different kinds of enzymes that we might have. And what catalysts did, they made these reactions happen quicker. So we can return back to this diagram that I had drawn here, showing this block being broken into two pieces. If this enzyme that we see here would be a catalyst, it would simply make this reaction happen quicker. And now that we have covered the very basic concepts on this chapter, we can actually move on to have a look of different kinds of macromolecules that we might have. So in this chapter, we covered carbohydrates, lipids, proteins, and nucleic acids. So we'll start with the carbs. So let's have a look of the carbohydrates. And I'm just going to try to highlight, oops, I didn't mean to do that. I'm going to try to highlight them a little bit more here. So on this chapter, we're having a look of, or this part of the chapter, we had a look of carbohydrates. And I think that many of us can think of many examples of carbohydrates. It's things like bread, potato, pasta, rice, corn, sugar, starches, all that good stuff that we often think of. But that's not the only kinds of carbs that we find. We also find vegetables, especially the green leafy vegetables that are excellent sources of carbs. So often we don't think about that, but those are really, really good sources of carbs as well. And carbs serve two functions. So the first function that we see is that they're a source of energy. So essentially what carbs are, they are sugars in a different number of units. So we have monomers, dimers, and polymers. In addition to energy being sugars, carbs also provide raw materials for the body to build many different things. And in this context, I do want to highlight an important term that I want you to be familiar with. And this term would be fiber. So what is dietary fiber? You may have heard, for example, certain muesli, certain granola, they're advertised as great sources of fiber, or certain types of bread. Well, fiber is actually something that you cannot digest. So we eat it, it goes into our body, but our body really is not able to absorb it and take it into the body to do anything out of it. It simply passes through. So it's the stuff that is non-digestible that passes through your alimentary canal and comes out from the other end. Why would we then eat something that we can't actually really utilize as such, absorb as such into our body? Well, there's few different functions. A diet that's rich in fiber gives us the feeling of being full. So when you eat and you get food into your stomach actually stretches. And I'm going to draw a really simple model of a stomach here. It's not the prettiest drawing I've ever done, but we'll live with that. So that's my imaginary, very simple, very crappy drawing of a stomach. But what happens when we eat food and it fills the stomach is that the stomach is stretched, its wall is stretched into various directions. And your body is able to feel this, and your body says that, okay, I'm full now, since my stomach is stretched into bigger than what it normally would be. So the fiber here helps us to feel full by doing this stretching, physical stretching of the stomach. And there's many examples of that being used. I think that back in the wild days, some models, for example, ate cotton balls and so on to stretch their digestive system so they would feel that they're full even though they're not consuming anything. Do not do that. That's very, very dangerous. You can actually end up developing a blockage into your digestive system. So that's not a good idea. The other thing why fiber is important for us, dietary fiber, why we want to consume dietary fiber, is that it helps to move materials along the digestive tract. And how it does is that it basically gives us this bulk that your body and your digestive system is able to push against. So it allows us in this way scrape the walls of the digestive system, so clean them, but also help the material to go through so you are more regular when you use bathroom. So that was the important two roles of the dietary fiber. I know that it's a little bit of a side angle to our discussion of the carbs, but it's an important concept to be aware of. And let's continue our journey looking what else do we need to know about carbs. Like I said, carbs could be made of a different number of units. And here we see first monosaccharides made of just one unit. If we have 2 units, we have a disaccharide and then a polysaccharide where we have more than 2 units, and the number could be however large at all. We end up seeing carbs that can be very, very, very long in structure. So that's the terminology that we want to know in terms of the carbohydrates and the number of units. And like you saw this single drawing that I've included here, that's basically just a single sugar's chemical formula where I have left out the other letters than just the oxygen molecule. And there is another side angle to this discussion that I included here. So you remember chitin was our example of a polysaccharide. And usually insects and all these kind of little bugs have their exoskeleton made of chitin. So chitin is this hard polymer of carbohydrates. And if you think of the metal harness of the medieval knights and so on, that would be a good example of what an exoskeleton looks like. So a lot of insects don't have an internal skeleton like we have. Their skeleton is actually outside them, and as they grow they might need to shed their exoskeleton so that it doesn't become too small and end up constricting them. So that was another side angle to our discussion of the carbs. There's one more concept about carbs that I want to discuss. So we can talk about having simple or complex carbs. And let's have a look of what's the difference between those two. And I'm just going to change the color so that it's consistent with what we did earlier on. So often people might wonder what's better, to eat simple carbs or complex carbs. And let's have a look of these examples of what simple carbs would be first. So things like candy, sodas, donuts, fries, all these kind of things that we often end up thinking about not being as good for us. Whereas the complex carbs, breads, vegetables, especially if you have multigrain bread, porridge, things like that, they are considered as complex carbs and considered as a preferred option of what kind of carbs to consume. So why? What is this that simple carbs are not as good as complex carbs? Let's think of that a little bit. So simple carbs are either 1 or 2 sugar molecules. So they're very, very tiny in terms of their molecular size. And this simple size, simple structure of them means that they're quickly taken into the body and quickly used. And because they are quickly available, and once we have used them, you end up with this sugar crash. So rather than having carbs that are very easily available for your body, we want to have carbs that are complex. So in this case, they would be made of multiple units. They would be polysaccharides. And by being polysaccharides, breaking them apart takes a longer time. And while it takes a longer time, this ensures that they're available in the body for a longer time as well, and available for when needed. So there's more steps involved with making them available, which is ideal in this setting. So we prefer to consume complex carbs because it takes a longer time to break them apart, and as it takes longer time, they remain available in the body for when they are actually needed. So that was our first example of macromolecules. And just for the making a little bit of a list here, let's add it here as the note, and we'll come back to these notes a little bit later, but we just want to list them for something that we're going to do later on. And apologies for my handwriting, I'm doing it on the computer, so it's not as pretty as normally. So that was our carbohydrates. Next, we're going to have a look of the next group of types of macromolecules, and these are going to be, in this case, are lipids. So let's discuss about lipids, what's important to know, why do we need to know about lipids, and what are the key things to know. So again, if we think of different kinds of lipids, it's easy to come up with examples of dietary lipids, oil, butter, cheese, lard, all that kind of good stuff again. But there are also other kinds of lipids that are considered a little bit better for you. So olive oil, fish, nuts, seeds, avocado. If you think, that sounds to me at least very much of a Mediterranean diet. So all those kind of good sources of fats. So if you want to make your life simple, you can consider lipids as simply as examples of fats. And your body does need some amount of fats, just the amount, how much, is what matters here. We don't want to go overboard, but some fats are needed. For example, certain vitamins, for you to be able to actually absorb them, they are fat soluble. If you don't have any fats in your body, they will not stick around. So fats and dietary fats are considered as excellent energy storage. They are a source of energy and help to store energy in a long term. So that's why we have developed fat tissue in an evolutionary sense. It's a very compact, very good way of storing energy in your tissues, in your body. And of course, we know that they do not dissolve into water, so they're insoluble. We also saw a couple of examples in our lecture materials that certain animals, for example, on top of their fur coat could have a fat layer, lipid layer, forming this protective layer. Also on the certain tree leaves, they have this waxy layer on top of them. So those were examples of where we might see lipids also in life being used with animals. And let's have a look of the structure of a single lipid. And lipid is made of the triglycerides. And triglyceride is made of one glycerol which is the one here on the chemical formula. So there was one of those. And in addition to that, we had our, I didn't mean to change that. Let's go back. And in addition to that, we had 3 fatty acid chains. So you'll notice these carbon atoms again that are connected to each other. So 3 fatty acid chains attached to a single glycerol made up our triglyceride structure, which we have described here. So that's the structure to be aware of when we talk about the triglyceride structure. What else should we know about lipids? These fatty acids could be different kinds. We had fatty acids that were simply connecting those carbon atoms with single bonds. So you remember there was just a single line connecting these, and that meant that they formed kind of this straight structure. There was no kink or nothing odd when we draw the chemical structure of these fatty acids. But then we had unsaturated fatty acids, where we did have, again, these carbons connected to each other, but then there was one or more double bonds between the carbon atoms. And every time that we had this double bond, it caused a kink in the structure, kind of this bend in the structure of a fatty acid. So these were our unsaturated fatty acids, whereas saturated ones were a straight line in terms of their structure. Unsaturated, they had this bend every time that there was a double bond. Well, why is this important? Why do we care? Why does it matter? All of those are very good questions in my mind. Well, we care because unsaturated fatty acids are normally originate from the plant sources. And they are typically liquid in room temperature. So we tend to find that, I'm just trying to change my highlighter to what I was looking for. So we tend to find out that unsaturated fatty acids originate from the plants, and they are, I didn't mean to do that, and they are liquidy in room temperature. That was considered as healthier kinds of fatty acids to consume. Whereas saturated fatty acids, they usually originate from animal sources, and they are solid in room temperature. And these are considered as not as good for the for your health. For example, increased cardiovascular disease risk with the saturated ones. So that's something to bear in mind. And then we discussed also in our lecture video a little bit about trans fats. And these are these imitation butters that you can see sometimes. I don't know the British, or I know the British brands, but I don't know the Americans. Things that we call there, for example, oh, it's so good, I can't believe that it's not butter. So these are when we have taken vegetable oils and added hydrogen to those to make them more solid, to make them appear as if they were saturated, even though they are unsaturated fatty acids. So taking something that's considered generally healthier and trying to make it appear in a room temperature as a saturated fatty acid chain. These are known as our trans fats, or hydrogenated fats is the term that I keep seeing used here in America a lot. Well, do we think that hydrogenated or trans fats are good? The theory or the idea behind sounded good to me, at least. But when we end up finding that these are not actually doing what we thought they would do, we thought that they would be a solution for health issues with the fats, we end up seeing that they actually are not quite as good for the health, and they're associated with number of health issues. So when you see hydrogenated fats, you might pay attention. They are usually largely used in cheaper products, products that are more cost effective to produce. But in long term, we at least based on the current understanding, they are not that good for your health. So I'll leave it to you to choose. Are you wanting to use margarine, for example? Or are you wanting to use actual real butter? And I had included a description of the manufacturing process of also hydrogenated fats in our full lecture video. So I'll leave it for you to have a look there if that's something that interested you, so you can jump back there. In terms of the lipids, we also want to talk about, and I'm just jumping back to see what color I was using. I was using blue. So let's jump back to the blue color. We want to also talk about phospholipids. And phospholipids are something that's going to become very important later on this course in the next couple of chapters, as I'm sure you've already seen, as phospholipids are the foundation of many membranes and plasma membranes that we end up seeing. And we'll discuss this concept time and time again, and the fact that we discuss it that often is probably an indication that they're gonna be important for you to know. So phospholipids, and like I said, we'll discuss them more, but at large they can be divided into two parts. Phospholipids have this head part, which is attracted to water, and because it's attracted to water, it's known as hydrophilic. It has an affinity to water, whereas the tail parts were hydrophobic. So what do you do if you have a phobia? You try to stay away from the source of that phobia, so you avoid the source of that phobia. So tails try to stay away from the sources or from the water. And we remember, and we'll see this concept a little later, that these phospholipids, because of the heads being attracted to the water, and because most of your body being made of the water, these heads are facing in your tissues towards the outside, towards wherever water is in your body, whereas the tails seem to spontaneously pack together so that they face against each other if we have two layers. So none of the tails have to be exposed to the water in this way. So this is something that spontaneously happens in nature. We don't really need to worry about it. It's just a characteristic of how phospholipids behave in water. Their heads face outside from the double layer because most of our body is water, and tails face inside, facing towards other tails, so they don't get exposed to the water at all. And the last concept about the lipids that we discussed and I want to highlight here was the concept of steroids. So a lot of hormones are made from steroids. They're the kind of basis for hormones. And steroids typically have a foreign chemical structure, And you can remember that your body does make a lot of steroids. You don't really need to get them from diet. High amount of sources from the diet are associated with an increased cardiovascular disease risk. So you don't need cholesterol from the diet. Your body produces all the cholesterol that you need. And I'm just going to jump back again to the beginning. Let's add our lipids to our list of different kinds of biological macromolecules that we're having a look of. And we're ready to move on to our next type of macromolecules, which is going to be proteins. So let's talk a little bit about proteins and why proteins are important macromolecule for our body. And some of the natural sources of proteins where we normally get proteins in diet, it's both the red and white meats, milk, beans. Also, if you think about it, egg whites are usually considered as a really good source of proteins. Tofu, if you're not a meat eater, fish, all that sort of things. And proteins have many uses. It's not just for building muscles, as we would often think about. We often think about these structural proteins, especially when you go to the gym, you see people taking protein supplements to make sure that they're able to grow their muscles as they exercise. But proteins serve a bunch of other functions as well. So the structural proteins are not the only group of proteins that we end up seeing. But the reason why you see some people taking protein supplements while they're exercising is that proteins from the diet are not stored in the body. So you must be getting them regularly from the diet so that you have the proteins available when you need them. And some of the people who exercise really heavily with the goal of growing muscles, they believe that it would be optimal to have as much proteins available when they're really putting in that workout or immediately after that workout. So that's the idea behind protein supplements. So what are proteins then? And we end up seeing proteins later on this course when we look at the structure of a plasma membrane and how there's proteins embedded in there. Well, proteins are polymers of amino acid. So if you think about it, proteins are just a bunch of amino acids connected to each other, and then they get folded into more complex shapes, and that's where these proteins that we often see in the plasma membranes that we're going to be having a look of later come from. And the structure of a protein, we have in an amino acid an amine group, carbonic acid group, and a side chain. And there's 20 different kinds of side chains that we can have one from. So there's a lot of variation in there. Then the next thing, and I think I mentioned this in our lecture video, that back in the day in one of the textbooks that I was using in my class, there was a picture of Nicole Kidman on the chapter when we talked about proteins, and the students were asking me that what does she have to do with proteins, and I quite frankly answered that I don't know, but I did some research, and at the time, and this I guess shows how long time I've been teaching, Nicole Kidman was probably the biggest star in Hollywood, and I think she had just gotten a perm. If I'm not mistaken, her hair is not normally straight, and this perm was a big deal, and that's why she was on on a textbook associated with the proteins. So hair is made of a protein, and if we change the chemical structure of a protein, the physical structure of a protein will also change. So, for example, when Nicole Kidman took a perm, she was chemically changing the protein structure of her hair, and that changed the physical appearance of her hair as well. So this just links not only that hair is made of protein, but the important concept that when we manipulate the chemical structure of substances, usually their physical appearance or their functioning also changes. So there's a link between chemistry and the physiology. Another example I think that we saw was the normal red blood cell, and then a single base difference that caused a sickle cell to be formed. If you are unfamiliar with that, I think that we covered that at least to a certain extent in our lecture video, so that might be something that you can also spot. So let's add proteins on our list of the macromolecules that we have covered. And there was a reason why I wanted to do this list. This list, and especially this order that we're seeing here, actually gives us the order of how your body prefers to use these different macromolecules. So ideally, your body wants to get carbohydrates from the diet. If there are carbohydrates as a source of energy in your diet, your body will consume them as a source of energy. However, if you go for a diet that's low carb or no carb diet, your body then goes to the next available source of energy, which is lipids, so fats. So this is why it's so hard to lose weight. If you have any carbs available in your body will consume them before it starts to consume the adipose tissue, the fat tissue. So this is the idea behind all these low carbs, no carbs diets, that if there's no carbs, your body will start to burn fat. And of course, if we don't have carbs and we don't have lipids, then your body will start consuming proteins. So an example of this would be, for example, when you go to the gym and you see someone who's very, very skinny, they don't have any fat in their body, they don't eat properly, they don't have carbs in their body. So what your body does then, if they don't have those two first options, it goes for the proteins. And the biggest source of proteins in your body, if you again don't eat sufficiently, is your muscles. Muscles are made of proteins. So your body can actually start to consume your own muscles to provide energy for that exercising. And that happens sometimes. So that's just something to be aware of when you're exercising. If you're exercising with a goal of bulking up, building muscle mass, you do want to have something that your body can build that from. So that was kind of a side note, but I did want to include this order of preference, how materials are used in your body. Let's have a look of what else we have. I think the fourth group that we had included, or I had included in these biological macromolecules, was our nucleic acids. So let's talk a little bit about nucleic acids, what they are and why they are important. So if you think of the DNA, RNA, the term Na there tells us, the Na stands for nucleic acids. There are different kinds of nucleic acids. DNA, of course, was stored our genetic information. And RNA is what is the copy that's made of the DNA and then travels outside the nucleus and can be used as a template for building proteins. We'll review this concept a little bit later. What I do want to review with you here, though, is the structure of a single nucleotide. So the basis of the of the DNA or RNA. In case of DNA, double strand, and in case of RNA, a single-stranded structure. So let's review that quickly. A single nucleotide is made of a single sugar that has a phosphate attached to it, and then a base. And sugar and phosphate are always similar, so we see the sugar-phosphate backbone of DNA and RNA strands. That's always the consistent part. It is the bases that vary, that are the ones that code the DNA code as such, if you wish. And we had different kinds of bases, and we see our potential bases here. So we have adenine, guanine, cytosine, and thymine in DNA. RNA is the same, with the exception that here, instead of thymine, we have uracil. So that's the difference in the bases between DNA and RNA. And you might remember that DNA was this double strand that was kind of a ladder that's twisted around itself, whereas RNA was just a single strand that exists as a template for the protein synthesis. So let's add that also to our list here as the last ones. Although we don't really use nucleotides as a source of energy as such, but that's there now just for the sake of that. And we'll talk a lot more about DNA and RNA in the few remaining chapters of our course, but I wanted to include them here as the basic macromolecules. And that completed our chapter 3. So we're ready to move on to chapter 4. If you need to take a break, this would be a great time to do so. If not, I'm just going to jump right into the next chapter, chapter 4, where we start to look at the cell structure. And this chapter is kind of a general review of different parts of the cell, whereas in the next chapter we focus in more detail on the cell membranes, the plasma membranes. And then moving forward, we have a look of different processes such as cellular respiration and photosynthesis, and so on and so on. Again, we start with some basic concepts. So the cell is described as the basic unit of life. So what does that mean, to be alive? Something that's an organism that's alive is capable of supporting itself. It does not really need other components to exist as part of its structure. So the cell is the smallest organism that's capable of sustaining life on itself. If you think about cellular organisms, for example, cell organisms, those little stuff inside the cell, well, they can't exist without other cell organelles. So they require the others, whereas cell is capable of existing by itself. And we find many single cellular organisms in biology that are just simple cell, exists on its own. On this chapter, we also talked a little bit about microscopy, and I have here a nice scale that shows what we can see with a naked human eye, and where do we need microscopes. And we learned that a lot of the study of cells kind of had to wait until we were able to advance what we can see beyond the naked human eye. So we needed to have the development of microscopes to advance before we were able to study different parts of the cell. And from the cell, you can probably with a naked eye see on most cells maybe the outline of the cell if you're really observant, you might be able to see the nucleus of the cell, but that's about it. However, with the development of light microscopes, we've been able to study the different parts inside the cell, so different cell organelles. And of course, as the technology developed, and instead of using light to look at the structures, we started using electrons that then bounce off from the structures. The electron microscopes developed, and we've been able to study all these smaller things than just cells. And study of atoms is kind of where we're pushing the boundaries and subatomic particles at the moment. But so to study cells, we could do that with both light microscopes and electron microscopes, a little bit maybe with naked human eye as well. So that was about the microscopy. So what we learned in this chapter was that the cells were always small in size. And the explanation that you were provided for the small size of the cells was related to this surface area to volume ratio. And I think that how it was explained to you was that when the size of the cell grows, so the volume of the cell grows, its relative surface area decreases. And what we mean by this is that we want cells to have a lot of surface area, a lot of this plasma membrane layer, the outermost layer of the cell. And once cell grows a lot bigger, the ratio between the volume of the cell and surface area of the cell decreases. So we're losing something that's really important for a cell to function ideally. So that's why we want to keep the cells small. And some of the explanations why it wasn't ideal to have large volume within the cell, we saw, for example, the transportation distances from the cell membrane to the inside of the cell grew too large. So we were no longer able to facilitate the passages of the material efficiently. The other concept that we saw was that a lot of the metabolic functions, so chemical reactions within the cell, took place on a cell membrane. So we also want, for that reason, to have plenty of cell membrane available. So we want to keep the amount of the surface area as large as possible in relation to the volume of the cell. There were a couple of techniques how we could increase the surface area as well, other than just keeping the cell small. We could develop different kinds of membrane folds or microvilli. So if this is the cell membrane, the outermost layer, by doing these folds, we're able to pack a lot of surface area in a small space. So that might be one way to get around this. Even if the cell grows in size, let's include a lot of the surface area by doing this. And later in your studies in biology, if you start looking up, for example, AMP, you'll notice that a lot of the structures where we want materials, for example, in digestive system, to be absorbed, there we end up finding a lot of these microvilli or membrane folds. So there's a lot of surface area for materials to cross inside the cells. What else? Well, there's some basic terminology that I wanted to mention and I want you to be familiar. So when we're studying cells, we talk about cytology. When we talk about study of tissues, that's histology. So just so you know what we're studying. We also discussed a little bit about the development of the first cell organelles. That's a question that students every now and then ask from me. And we have a generally well-agreed hypothesis of how the first cell organelles appeared, and we think that they were accidentally ended up inside the cell, another organism that by accident ended inside the cell and ended up forming a symbiosis with the cell. So both parties benefited, or at least one party benefited, another party wasn't harmed. So as a result of that, slowly cell organelles became more and more common, and we started to see them in the cells. And we also talked about two main categories of cells that we could find in the nature. And the first one that we had a look of were prokaryotes. And we saw that prokaryotes were very simple, small cells. They were the ones that evolved first. And like we mentioned, they're small in size, simple in terms of their structure. There's very little anything else than the cell membrane, maybe some ribosomes floating in there, and the DNA is floating free in the cytoplasm. There's no nucleus, so it's just floating in there. That's all that a prokaryote is. Whereas eukaryotes are much more larger. They developed later in life. They're much more complex also in terms of their structure. So we end up finding a nucleus. That's another membrane enclosed organelle, and the DNA is stored inside there. There's many other cell organelles that we end up finding within the eukaryotic cells. So a lot more structures in there, and a lot more happening in terms of the physiology as well. So eukaryotic cells took longer to appear. They're generally more complex, and what's striking about them that they have these various different compartments, various different structures inside them. So they're much more varied in their structure as well. So you want to be familiar with the similarities and differences between the prokaryotes and eukaryotes. So just quickly as a review again, prokaryotes, they're first ones to evolve, smaller in size, much simpler, and the DNA is just floating free in the cytoplasm. We don't end up finding a nucleus in the prokaryotes. Whereas then our eukaryotes, instead, we notice that it took longer for them to appear in life, so evolved later. They're much larger, much more complex. Also, we end up finding these separate compartments within the eukaryotic cells, so all these cell organelles that we had a look here. And of course, the DNA was housed inside a nucleus, a structure on its own. So that's the division in two that we want to be familiar with. What else? We then started to review the different cell organelles, one organelle at the time. And let's do just that. And the first cell organelle that we're having a look of is when we enter to the cell from the outermost layer, and that's going to be, of course, our plasma membrane, sometimes also described as the phospholipid bilayer. So that's the cell membrane. And you remember the phospholipid bilayer, that term referred to the fact that these phospholipids, which had the head parts that were attracted to the water, and then they had these tail parts that wanted to avoid the water. Well, when you put a bunch of them floating around, and you remember that most of the human body is actually made of water, they want to organize in a way where all parts of the phospholipid are happy. And the easiest way to organize in this way is to form this phospholipid bilayer structure. So this is what our cell membranes look like. So the heads are facing outside, so facing towards water, and tails are facing inside. So that's just how spontaneously in the nature the phospholipid bilayers are formed. Plasma membranes are also described sometimes as fluid mosaics with this fluid mosaic model. And let's have a look at what that means. And let's start with the term fluid. Well, the fluid refers to the fact that actually the plasma membranes that we see on the textbooks are not really good representation what they are like. They're more of like snapshots of a single time. In reality, plasma membranes are constantly changing in their structure. So all of the parts of the plasma membrane are constantly moving about, changing sides, changing their location. In addition, many materials are constantly moving across the plasma membrane. So it really isn't as stationary structure as we often end up thinking. The other term that we see there is the mosaic. Well, anyone who has seen mosaic art knows that it's made of many different pieces, usually of a ceramic or stone, that are put together to form a bigger picture. Well, it refers that it's made of many different components. So in case of the plasma membrane. So fluid mosaic model is often used to describe the plasma membrane structure. And that is a kind of an important concept. So make sure you're familiar with that. If based on this description you're still lost, I totally encourage you to go back to the lecture video to have a look of that. We have already earlier on had a look of the general structure of a phospholipid, but just in case if you are unfamiliar with that or have forgotten, let's just review it very quickly together. So we have the phosphate group here. That's the first part that we want to know of a phospholipid. And I'm just trying to, if you're wondering what I'm clicking away, I'm trying to match the colors so it'd be easier for you to follow. We'll see how successful I'll be on that. In addition, a phospholipid contains also our glycerol group here. And again, let's see if we can highlight that with the correct color. And finally, in a phospholipid, we also find two fatty acid tails. So here's the first one and the second one. And notice the double bond on the second one that causes this slight kink that we might end up seeing. So you might be wondering that, okay, now we're writing the chemical structure of the phospholipid, but earlier on I showed you a different kind of a drawing of a phospholipid. I showed you that a phospholipid looked like this here. It had a head and it had tails. Well, this is the exact same thing. As you see below, I just simply have written out here the chemical structure. So the phosphate and glycerol form the head part. And the tails were formed by two fatty acid chains. And we already discussed earlier on how the heads were hydrophilic, so they were attracted to the water, and how the tails were hydrophobic, so they were afraid of the water, if you wish. They wanted to avoid water to their best ability. So this concept should be familiar as well from what we have already discussed earlier on. Again, you notice that certain terms, certain themes keep coming up time and time again. That's usually an indication that that might be an important concept to be familiar with. And here we see something that we just actually have discussed already earlier amongst ourselves. So these phospholipid bilayers. So notice the term bi here, telling us that there's 2 layers, 1 and 2, and these layers are made of phospholipids, so a single phospholipid here, and we just discussed about what a phospholipid was over here, so if you want to review that. So what does the phospholipid bilayer tell us as a term? It tells us that a phospholipid bilayer is made of 2 layers of phospholipids. So 1 layer and 2nd layer. And their heads were facing outwards. You remember here. And tails were facing inwards. And this way, because most of the surroundings, most of our body is made of water, everyone's happy. The heads get to face towards the water, which is both outside and inside the cell, and the tails get to stay away from it to the best of their ability. So everyone wins, everyone's happy. That's what we want in life, right? And then we discussed a little bit about different folds with different microvilli. Their function really was to increase the surface area if there was a need for that. We remember that we had normally small cells because we want to have enough of the cell membrane surface area. That's usually important for us. The next group that we discussed were proteins. And I'm going to use a different color to highlight them, talk a little bit about proteins with you guys. I know that we've mentioned proteins in 2 different concepts. Now we're looking at the proteins within the plasma membrane. So proteins, if you look at this diagram that we've drawn before, we're seeing it here at the top right corner of the screen, but I'm actually going to draw it again. So we saw our phospholipids here, typically the heads facing outside and tails facing inside, right? That was the typical arrangement of a phospholipid bilayer, and that made sure that the heads that had an affinity towards the water were able to fulfill their goal, and the tails that were hydrophobic, wanted to avoid water, could do so. They managed to avoid water. Everyone was happy. Everyone reached their goal, and this is how these plasma membranes spontaneously arranged in nature most of the time. Well, proteins in all of this, let's see how they fit in. We remember that we discussed about the plasma membranes being mosaic in their structure. So often within the plasma membrane structure, we also end up seeing that there's proteins embedded there every now and then. And proteins have a huge bunch of functions, as you see listed there. They can be channels, so facilitate the movement through a plasma membrane. So they might be like tubes for material that's too big to sneak in through between these phospholipids, or if we want to have a protein that pumps the substances across the plasma membrane to one direction but not to the other, or so they would transport things, or what else could they do? They could be involved in recognizing, for example, what's a cell from your own body, what's a foreign cell. So that's an important one in terms of recognizing pathogens, but also can dysfunction when we think about, for example, organ rejection. Can also recognize, for example, hormones, that this hormone should act on this cell. So they can be receptors. So receptors only act with certain substances. They need to be a match, like a lock and a key. Or they can function like enzymes. What did enzymes do? If you remember, this would be a good time to test yourself. So they assisted in chemical reactions. Proteins can act like an enzyme. Proteins can also work as junctions between different cells. And we'll see a couple of examples of that right at the end of the chapter as well. So proteins were the second cell part that we discussed. Then the third cell part that we want to discuss is going to be our cytoplasm. So cytoplasm is all other material inside the cell. So kind of this clear or clearish jelly-like fluid that fills the cell. So if you have the cell that we drew earlier on, so we had the cell membrane, we had a nucleus, and then we have a bunch of organelles there. So cytoplasm in this case, I didn't mean to change the color of that. Let me just change the pencil color. Cytoplasm is all that liquid inside the cell, and it includes all these cell organelles with the exception of the nucleus. Nucleus is not part of the cytoplasm, but everything else within the cell would be. So that's our cytoplasm. So if you think, why do we have cytoplasm? Well, first of all, it fills the cell. We can't just have cell empty because there needs to be something in there. And by having this fluid, jelly-like fluid, it also helps to keep the organelles in their place. So it fills up the gaps, helps stuff to stay where they should be inside the cell. So that was our cytoplasm. And next we should talk a couple of words about the nucleus. So as you remember, nucleus was this central structure within the cell. We typically see it in the center of the cell. When we stain cells, we see that it usually stains dark. That's the dark blob inside the cell. And that's where the genetic material of the individual is stored. So DNA is within the nucleus. So that's also something to remember. What else can we say? Well, nucleus had a double membrane envelope. We called that nuclear envelope, and within that envelope it had small holes. Those are known as nuclear pores. So the function of the function of the nuclear pores was to allow certain materials to enter into the nucleus and other materials to leave from the nucleus. So just like other parts, certain things need to get in and other things need to get out. And since it's this double membrane, this was made possible by having these tiny holes known as nuclear pores on the envelope. Within the nucleus, like we said, was DNA, and a section of a DNA that codes for a specific protein is known as a gene. So a part of a DNA that codes for a functional protein is the gene. And there's a lot of material in DNA that actually doesn't code for anything as well. The term chromatin, that should be also something that you remember from our chapter material. That's made of protein, DNA, and RNA. So if you have forgotten that, again, jump back and have a look at that lecture video. What other things have we discussed? This is something that we discuss in this stage, but it will also become important a little later on. So this is an important concept for you to be familiar with. And let's have a look of how DNA gets turned into protein. So DNA was this two strands that were intertwined, almost like a step ladder that had been bended so that the bases were these horizontal lines. And DNA never leaves the nucleus. It always has to stay within this nucleus that we're seeing on this diagram here. What is capable of leaving nucleus, though, is the RNA. So you can consider DNA, I think I used this as an example, as a recipe book. DNA contains recipes for different kinds of proteins. But when you're about to bake, no one takes the entire recipe book and cook every single recipe that has ever existed in that cookery book. That would be just silly. What we do instead, we take the recipe that we need at that moment to make that dish, or in this case, to make the protein that we want to make. Well, this DNA is now like a recipe book that's in a library, and the library would be analog now to the nucleus. You are not allowed to check out that recipe book. Instead, what you can do, you can make a copy of it, whether it's a photocopy or handwritten notes. You can make a copy of the DNA, and this copy is known as RNA, of course. And RNA can travel out of the nucleus, and it travels to the cytoplasm, and it travels to the structure known as ribosome. What a ribosome is the cell organelle that actually makes proteins. So in this diagram, ribosome will be made of one larger subunit and one smaller subunit. And DNA goes through it, and with each three bases of or RNA goes through it, and with each three bases of an RNA, we bring in an amino acid. So these are individual amino acids. And as we find a match for an amino acid that matches with three bases in the RNA, it gets added to this chain that we see here. And this chain put all together, I'm having trouble getting to draw there. This chain put all together is gonna be our protein. Then it folds and does all sorts of other things. So notice DNA never leaves the nucleus, whereas then we saw that the RNA made a copy of the DNA, and that was to be found both in nucleus and outside the nucleus. We'll discuss more about the concepts of, and I wonder if I can do this with this, let's see, concepts of transcription and translation later on this course, but at this point I do want you to know what stays within the nucleus and what does not. Okay, the next cell structure, we've got a lot that we're going to go through on this chapter. We've got mitochondria. Well, for the longest of time, we have taught students, and I think you might have heard in your high school classes or other courses, that mitochondria is the powerhouse of the cell, or cellular powerhouse. So that refers to the fact that the mitochondria makes ATP, which is the chemical form of energy in the cell. And that is true. We said for the longest of time that mitochondria was the only part of the cell where energy was produced. Well, we're finding out that that's not quite the case. Energy, ATP, can be produced in few other places, but the majority of the ATP in the cell is produced within the mitochondria. So what mitochondria does, it takes carbohydrates, so sugars and oxygen, and makes energy out of them. As a byproduct, we are left with carbon dioxide and water. So if you think this is all that your body does, this is what being alive is all about. You take sugars, you need oxygen, and that makes energy. As a waste product, carbon dioxide and water. So this all happens at the level of a single cell, at the level of a mitochondria. So just like you need to get sugars, you need to get energy into your body, and just like you need to get oxygen into your body, cell needs those as well. So we see this all in a much smaller scale than you might have thought earlier on. Making the energy, making ATP is the goal of the mitochondria. And a couple of terms that we're seeing there, cristae were the folds inside the mitochondria. So that was the term that we used to describe those. I'm going to try to draw it here. This is probably going to look really messy because drawing on a computer is not my strongest field. But mitochondria is often this kind of a sausage-shaped structure. And inside it, it has this outer membrane where we have all these folds. And these folds inside there are these cristae that we end up seeing. And a lot of the processes when we are producing energy take place within these cristae. So maybe not the fanciest drawing I've ever done, but you've got much better pictures on your lecture material. But that should give you an idea of what we're talking about when we're talking about the cristae. So they're these little folds. Another interesting thing that we noticed when we talked about the mitochondria was that it actually has its own DNA, and you inherited that mitochondrial DNA only from your mother. So that had some consequences in a forensic applications. And like I shared with you, I've been doing a bit of work on the forensic field in the past as well. So that's a rather interesting side angle to this story. Then we should have a look of the chloroplast. So chloroplast is only found in plants and algae. So we don't find chloroplast in animal cells. We humans are an example of animals. We cannot do photosynthesis. We need plants. We rely on plants for source of oxygen and source of sugars for us to be able to function. So only plants and algae can do photosynthesis. They have chloroplasts in their cells. So what happens in photosynthesis? Let's have a look of that a little bit. In photosynthesis, we take solar energy, so sunlight, and in addition to that, we also have water and we have carbon dioxide. So these three are all the things that your plants need. Your plant needs some sunlight, it needs some water, and it also needs some carbon dioxide. This is why when you sometimes see people who say that, oh, my roses do so well, for example, because I speak to my plants. It's not really speaking to your plants that make them do so well. It's the breath that comes out of your mouth that's rich in carbon dioxide that the plants benefit from. Well, So what do these plants do with all this? They make sugars. So this is chemical formula for a simple glucose molecule, and then as a byproduct they also make oxygen, which happens to be very convenient for us humans and other animals. We rely on plants for the oxygen. So that's what chloroplasts do. And this kind of takes us to something that we will talk later on this course. I just think that in the interest of time, we won't spend too much time on that. We'll notice that this formula that we have for photosynthesis is an opposite of that of the cellular respiration. So it's almost the same formula, just the other way around. Plants in photosynthesis make sugars. Animals then use sugars for energy. So same formula, just the other way around. And when we looked at a structure of a chloroplast, we saw that they were this double membrane structure, and inside there was this aqueous fluid stroma. There were also these disc-like structures, and multiple of these disc-like structures, when they're piled together on top of each other, each pile is known as granum. So we end up seeing these inside the chloroplasts. And again, your lecture video covers the structure of a chloroplast in greater detail, but I did want to mention those structures because they are worth of knowing for you. So no stroma, the fluid inside the chloroplast. Thylakoid was one of these single disc-like structures that we saw, and then granums were a pile of these thylakoids on top of each other. Next structure that we're going to talk about is going to be our endoplasmic reticulum. That's quite a mouthful, I think, and really, to be honest, we don't often write it out. In my field, we just abbreviate it as ER. So this is this network of continuous membrane tubes, loops, and they often are connected with the nucleus's nuclear envelope as well. And there were two kinds of endoplasmic reticulum, smooth and rough ER. And if I just again draw a very, very, very simple demonstration, if this is our nucleus, and from there we had this ER coming out, it's this structure of various different membranes that are connected. Some of it is known as smooth ER. The smooth ER has nothing on its membrane structure. It literally is smooth, and there's many functions that smooth ER has. And again, refer back to your lecture material for a full listing of those. But what I wanted to highlight here, that some of the ER then have ribosomes attached to this membrane. And this is known as rough ER. These ribosomes, of course, you remember their function was to make protein. So I just want you to be familiar to separate between the smooth and rough ER. So that should be something that I hope you are familiar with. If not, again, refer back to the lecture material. The next structure that we're going to talk about a little bit is going to be Golgi apparatus. This is a stack of these folded membranes, if you wish, and also kind of vesicles within the cytoplasm, again, I'm trying to draw it. It's probably not going to be the best drawing that I've done, so you have a better picture on your lecture material. But it looks a little bit like these membranes that are piled together and kind of hug into each other, if you wish. And what Golgi apparatus does, I compared it during our lecture video into a distribution center of a cell. So Golgi apparatus receives materials from other parts of the cell, often through various different kinds of vesicles. This material then is processed within the Golgi apparatus. It might be repacked, reorganized, resorted, and then it's sent out, usually in a vesicle, to go wherever it needs to go. So really, all of those would be functions of a distribution center if you have ever dealt with logistics. That's basically what Golgi apparatus does and what we can be happy at this level of the studying of the cell organelles. Then we had various different vesicles. These were these little sacs that transported material within the cells. Lysosomes they were sacs that contained digestive enzymes, so they help with breaking down stuff. And vacuoles, we often see them in plants. They are these large membrane stacks for storing materials, and a lot of the time they can actually take up even most of the plant cells' size. So if we have a cell here, we have a nucleus here, and we have a bunch of other structures there that do things, but the vacuole can grow to be size of most of the plant cell. So that's something that we can sometimes end up seeing, especially, like I said, in case of plant cells. So that's what vacuoles are. So I'm starting to color. Okay, a few other things that we see within the cells, and this is something that's going to become much more familiar as we continue and we start talking about cell division, DNA, and so on. Centrioles, they're a pair of cylinder tubes, and they're located usually close to the nucleus, and they really play a role when the cell divides. So at that time, we'll have a look of the centrioles when we address that a little bit more. And when you think of the cell, we saw the cell drawing. I've done a few different drawings, but here, for example, this eukaryotic cell. It's not just the cytoplasm that we have filling the insides of the cell. In addition, we find cytoskeleton inside the cell. And cytoskeleton is this bunch of different kinds of filaments, different kinds of tubules within the cytoplasm. If you think of their structure, you can consider them as working as the scaffolding of the cell. Again, I'm just drawing a very simple diagram of the cell. So what does scaffolding do? Scaffolding gives us not only physical structure. If anyone, for example, has worked at the construction sites, you see that there's scaffolding there. It helps stuff to stay on place, for example, if we need to climb up the walls or to the roof. So that gives the cell its structure. But in addition, materials can move along the cytoskeleton. It's almost like highways or paths for materials within the cell to travel as well. So that's the function of the cytoskeleton. And the final concept that I wanted to cover on this chapter was the various different kinds of cell junctions. And I grant that these are rather small diagrams, the full-size diagrams are again in your lecture material, but you can see a little bit of the 3 different kinds of cell junctions. So here we have 2 cells joining together. Tight junctions, if you wish, they're kind of these zipper-like bands connecting the one cell to another one. Whereas desmosomes, or sometimes these are also known as adhesion junctions, they have these large spots on both sides, and from these we have these various filaments that run out, connecting inside of both of the cells. And finally, gap junctions, that doesn't show up that well on this diagram, but they are these kind of channel-like lines, structures, protein-like junctions connecting from one cell to another, facilitating the movement of the materials from one cell to another really well on this case. So I would be able to again recognize each one of these based on the diagrams that you have been provided in your lecture material and also on this review sheet. And the last chapter that we discussed was the chapter on the plasma membrane. So I'm going to try to again pick a different color to do highlights a little bit. And you notice that this chapter had a lot of overlap with what we already discussed in the last chapter. So it kind of expands and continues of that discussion. And plasma membrane is really an important structure for us. Of course, we talk about cell membrane. We've already discussed about phospholipid bilayer. That's another name for it. We already discussed about how it is a fluid mosaic. Selective permeability means that some materials can enter and other materials can exit, but other materials cannot enter and some materials cannot exit the cell. So plasma membrane is also selective in that sense. So it's really an important structure if we think of cell's function. And we talked a little bit about the history of the discovery or definition of the plasma membranes. We noticed that it took quite a lot of time for us to have the machinery that allowed us to study the plasma membrane in greater detail. But to me, early on the 1900s, we already knew that there were lipids and proteins that made up the plasma membrane. The model of the railroad track, which was proposed by Davidson and Danieli, was that, okay, they said there's going to be 2 layers, and then there's almost like these sleeper posts that we have on a railroad that connect these 2 sides together. What Davidson and Danieli kind of missed, where they were wrong, they were saying that these railroad tracks as such, these metal tracks, would be proteins, and it would be phospholipids that go across a double layer of proteins. Of course, we know that this is not the case. We actually now know that rather what we have, I'm just trying to change the pen color if you're wondering why I'm clicking around. What we know rather that they had it wrong way around, that these metal tracks that they talked about, these are actually phospholipids, not the proteins. So this is the phospholipid bilayer structure, where we had the heads facing outside, tails facing inside. So Davidson and Danieli just had that wrong way around, but that's what they were. And then we also know, of course, that what goes through the plasma membrane, that was going to be our proteins. So if That's not the prettiest drawing, but if that helps you to notice that. But to me, it's quite striking that already earlier on, we had a fairly good idea of what's happening with the plasma membrane. And then later on, we have already, the fluid mosaic model was introduced, and we have already talked about that. So this should be revision. So again, we remember that the part mosaic refer to the fact that it's made of many different components. I've listed some examples there, of course, phospholipids and proteins, but also carbohydrates, cholesterols, and so on. And the part that is fluid refer to the fact that it's constantly changing, also materials moving across. So this should be revision if you want to check that out. Be familiar with that since it keeps coming up that often. We've already seen this phospholipid structure before, how we had our phosphate group that we find in a phospholipid. We saw also how we had a glycerol. Let's see if we're able to highlight that there. Oh, I meant to do a different color. And then we, of course, had 2 fatty acid chains within the phospholipid as well. So that was the chemical structure of a phospholipid, if you wish. And now that we have looked at this chemical structure, we will quickly notice that it looks very much like this diagram that we have here. We have the head part and we have the 2 tails. So if we want to fill in, again, we have already discussed about this as well, but if we want to fill in to our chemical formula, we saw that the phosphate and glycerol did the head part, and the tails were the fatty acids that we had. And then when we had a look at the fatty acids, this is again revision. We remember that we had saturated fatty acids. On saturated fatty acids, it's single bonds between the carbon atoms, whereas on unsaturated fatty acids, we had 1 or more double bonds. And this double bond every time caused this kink or bend in the structure, physical structure of this fatty acid. So we've already discussed again also about the hydrophobic and hydrophilic ends of a phospholipid. Hydrophilic, the heads attracted to the water. Hydrophobic avoided the water, the tails. We also discussed about the phospholipid bilayer, the concept, how they spontaneously arranged themselves into 2 layers. So by 2 layers made of phospholipids, just because of the characteristics, the nature of the different parts of a phospholipid. And we find phospholipid bilayers, of course, as the outer layer of the cell membrane, that forms the outside of the cell, but also around cell organelles. It was this that on eukaryotic cells, we found these compartments within the cell as well. And that's thanks to these cell membranes. Then we have already talked a fair bit about the proteins, but we're going to take that and expand that a little bit from what we have already discussed. So it's a little bit of revision, a little bit of new stuff. So proteins, we've seen them already earlier on within the structure of a plasma membrane, as if these proteins that go through the entire distance of the plasma membrane. Well, these proteins that extend all the way through a membrane are known as integral proteins. So they go through fully the entire distance of the membrane. We can also have other kinds of proteins, these proteins that go through only partially to the plasma membrane. So these proteins are known as peripheral, so since they don't go through the entire membrane. They typically are found only on one or another side, so they're not that commonly found. I'm again trying to get the just colors work so we can do a little bit of color coding. Some students learn better with that, some don't. So if it doesn't work for you, don't worry about it. I'm just trying to give different ways how you can learn these things. So peripherals were only partially through the plasma membrane. They're not through the entire distance. So that's what I wanted to say about proteins on that part, and then we had a look of the levels of structure on the proteins. So if you remember, we had primary, secondary, and tertiary structures. On primary structure, it was just the amino acid sequence. On secondary structure, we started to see these pleated sheets when we were studying the structure of a protein. And on the tertiary structure, we saw this kind of a tube-like model that's kind of, again, stepping further away, giving us the global view of the protein. So we can study it at many levels of detail. And again, we see this thing about Nicole Kidman's hair. I do think that that would make a really good extra credit question. And again, there I would require you to tell not just that hair is protein, but discussing about this concept that if we change the chemical structure of a protein, as she did with doing the perm, that's chemistry changes the chemical structure of the protein, that will also change the physical structure, the physical appearance of that molecule. So that was very visible on the outcome of that chemical reaction on her. Then we had a look of different kinds of transport processes through the membranes. And the first ones that we're going to cover are going to be examples of passive transport. So when we have passive transport, what happens is that there is no energy invested in that. So passive transport just happens. We don't have to spend energy resources to make that happen. And usually that's because of the concentration gradient difference. So materials move from areas where there's a high amount of material, so high concentration of something, they move towards the areas of low amount of the material until equilibrium is reached, so there's the same concentration on both sides of the membrane. And the first example of that we had a look of was diffusion. And what we see in diffusion is that initially we find that we have these molecules on one side of the membrane. So we end up finding that there's a high concentration on this side of the membrane and low concentration on the other side. And we saw that in diffusion, the materials moved through the membrane from the area of high concentration to the area of low concentration. And this keeps happening until the amount of substance is the same on both sides. So in this case, until we would see 4 balls on both sides. Now, the concentration is same on both sides. So we have reached an equilibrium, and that's how diffusion works. So these substances that are shown on this diagram as these red dots, they're able to cross the membrane, and they do that until there's an equal concentration on both sides. So that's what happens in diffusion. We aim to work passively thanks to the difference in concentration gradient until there's the same amount of substances on both sides. That just happens. There's no energy needed to put into it. Stuff just moves across this membrane that we're seeing here, right in the middle of the picture. I'm just trying to highlight the membrane in case anyone's forgotten that that's our membrane. Okay, let's have a look at the next example of passive transport. The next example of passive transport that we had was process of osmosis. In osmosis, again, we have these molecules here, and there's a greater amount of them on one side than there is on the other side. So we have a high concentration here and a low concentration here. However, this time what we end up noticing is the challenge with the fact that these molecules themselves, these red dots, cannot cross the membrane. They are prevented from crossing the membrane. However, yet we want to have a same amount of dots on both sides. You notice that there's only 2 of those here initially. We don't pay attention to this one. So how can we make sure that there's the same concentration on both sides? Well, the way to do that is that rather than these red dots moving, let's move water from one side to another. And we do that, and the water moves actually higher on this side where there's a high concentration of it, of the substance, and it moves lower on this side where there's less of it. So if we only have a little amount of water and solutes in it, and then we have a large amount of water and solutes in it, now their concentrations are actually the same. So we again aim for the equal concentration, but this time it's gonna be the water that moves from one side to another. I don't know if the color that I picked was the best for the purposes that we were trying to illustrate. So I'm just adding a little bit of darker color to show that the water moves from one side to another. So the water level climbs up on the area where there's a high concentration, and it decreases on the area where there's a low concentration. This allows us to have same concentration on both sides of the membrane. So again, at the end of the day, the concentrations are the same until there is an equilibrium. But in case of osmosis, it wasn't the substance as it was in diffusion that moved across. In these cases, it was the water until we got same concentration of both sides. And this gives us a basis for something that's going to be important when you go to clinical practice, and the concept is going to be tonicity. And if you remember, I had a bit of an example of that. I talked about different kinds of cells, and I think I had some silly mnemonics that I was using there. So let's have a look of different kinds of tonicity. And the first one that we're going to have a look of is going to be hypotonic cells. What happens in hypotonic cells is that water flows inside the cell, because the inside of the cell is more salty, and again we try to balance off this concentration difference. Well, we balance it off by moving water into the cell, as we can see here. Water is traveling in to the cell, and as a result of that, the cell swells. It becomes bigger and bigger and bigger. And in an extreme case, what can happen is that the cell can actually end up bursting because so much water is flowing in there. So